This video is about the electron transport chain. In it we will give you a basic run-through of how this energy powerhouse works. The first thing you can see drawn is the mitochondria. This is what houses the ETC or electron transport chain. The first enzyme is the NADH dehydrogenase. The little circle drawn here is known as the Q shuttle. It shuttles electrons in between the enzymes. Feel it like a ligase. This second enzyme is the BC1 complex. A couple more phospholipids. And a C shuttle, similar to the Q shuttle. The third enzyme is the cytochrome oxidase complex. These chains being drawn are made up of tiny phospholipids which separate the intermembrane space and the matrix. And the fourth, and our personal favorite enzyme, is the ATP synthase. Now you can compare our small diagram of the mitochondria in the top right corner with the cross-section of one small part, the giant thing really. The way that the ETC works is by shuttling electrons through the phospholipid bilayer to activate the enzymes, which works as pumps. These pumps shoot protons through to the other side to create a proton gradient, as you will see soon. The first electrons are taken from NADH to activate the first enzyme. These electrons are then shuttled over by Q shuttle to the BC1 complex, as seen here. This pumps another H+ proton through. They are then shuttled to the cytochrome oxidase complex by the C shuttle, which pumps another H+ proton through. This is the proton gradient. The second set of electrons are taken from the FADH2 by the Q shuttle. They keep getting shuttled through each enzyme until the cytochrome oxidase complex, when they are taken by oxygen and an H+ proton and create H2O, or water, as most know it. Right there. See they touch there, like that, to create water. Now, the hydrogen protons that are pumped through then travel to the ATP synthase. They are pumped through the enzyme and activated so that it can catalyze a reaction between ADP and a phosphate. This creates ATP. As you can see, there are 5 H+ protons out there. That means that each glucose molecule can make a total of 5 ATP. There's 2, there's 3, 4, and the last one to make 5. Now, let's run through that once more just to give you a general idea. So the first electrons taken from NADH, second set are taken from FADH, which also pumps another H+ proton through, right there. And then 2 more come through this one. Then they are taken by water. They go through ATP. And there you go. There's the equation for it all. C4 plants have a special way of carbon fixing as they live in hot and dry climates. They do this to minimize photorespiration. First, we will show you what a C4 leaf looks like on the inside. These small bubbles are known as the vascular tissue. They transport the sugar. These larger bubbles, drawn here, are the bundle sheath cells. And these final bubbles that are being drawn right here are the mesoville cells. And the rest, as you can see him drawing, will be labeled. That's their stomata, and those are air spaces. Now let's take a closer look at the individual cells. The first large green cell is the mesoville cell. The middle cell is the bundle sheath cell, and the final cell is vascular tissue. The cycle being drawn here is used in the end to create carbon dioxide for the Calvin cycle through malate. Carbon dioxide is put into malate so that it can be moved into the bundle sheath cells from the mesoville cells. And then, through the Calvin cycle, comes a sugar, which is given to the vascular tissue to be moved about the plant. Smiley faces, don't know why they're there. And there we go. Now, he's tracing the route that the carbon dioxide would take to get to its final destination, the sugar. Now, on to CAM plants. CAM stands for crassulacean acid metabolism. CAM plants differ from C4 plants because they regulate when each cycle takes place. Specifically that the C4 cycle takes place at night, while the Calvin cycle takes place during the day. This greatly minimizes photorespiration. As you can see, At night, the somata opens and CO2 is converted to malate, and during the day, the somata is closed and malate releases CO2 into the Calvin cycle. Now, we will be giving you a more in-depth description of the Calvin cycle and how it works. The three molecules that are being drawn are 3-phosphoglycerate, G3P, and rubapa, which is needed to start the cycle in the first place. The first thing to be inputted is carbon dioxide. There are three phases in the Calvin cycle. The first phase is carbon fixation. Carbon fixation is the conversion of carbon from an inorganic to an organic form. The second phase is reduction. In this phase, each molecule of 3-phosphoglycerate gets another phosphate. Also in this phase, NADPH becomes NAP+, and ATP becomes ADP. The third and final phase is regeneration. This process rearranges the molecules so that the entire cycle can restart. And now on to current events, which we decided to do on Ebola because that is very current and is an event. It affects the ETC because it attaches vital organs, which is like mitochondria, and the ETC does not receive proper amounts of NADH and FEDH from glucose, which is lack of eating, and ATP is not made as much, causing a lack of energy, and basically because you have no energy, you die. What do we do about it? Cure Ebola, don't get Ebola, or don't come in contact with anyone with Ebola. And that concludes our video. Thanks for watching, and stay classy, San Diego.